Hello everyone, welcome back to our channel. In today's video, we are going to explore one of the most fundamental and important resources in bioinformatics, that is the nucleotide database of NCBI. If you are a student or a researcher or someone who is curious about DNA and RNA sequences and their applications in bioinformatics, then this video is for you. Without any further delay, let's start today's video. We are going to discuss about three major questions, that is, what is the nucleotide database of NCBI? Second is, what does it contain and how to access this nucleotide database, and how to use the information present in the nucleotide database of NCBI. Let's answer the first question. So as the name indicates, nucleotide database, of course, it contains DNA and RNA sequences. Rather, we can say that it is a giant hub of DNA and RNA sequences. You can think of it as a giant library which contains nucleotide sequences instead of books. The second question is, what does it contain? Of course, it contains DNA and RNA sequences. However, there are three major resources of these nucleotide sequences in the nucleotide database of NCBI. The first is Genbank. Second one is ref sequence, which is popularly known as ref SCQ, and the third one is DDBJ or EMBL. Genbank is basically a database which stores all the DNA and RNA sequences submitted or deposited by the researchers community across the globe. Second one is the ref sequence or ref SCQ, which stores the reference sequences. So it is basically a large pool of reference sequences. Third one is DDBJ or EMBL, which are repositories and which contains globally connected sequence pool. So overall, we can say that NCBI Nucleotide Database is a large pool of globally connected nucleotide sequences which contains either reference sequences or experimental DNA and RNA sequences deposited by the researcher community. The third question is how to access the nucleotide database. For that, let's move on to our computer screen. Now let's do a very quick hands-on demonstration on how to use the nucleotide database of NCBI. For this, open your browser and type NCBI and click on the very first link, that is National Center for Biotechnology Information. Clicking on this link will lead to the homepage of NCBI, and this homepage will contains a drop-down menu. As you can see here, this drop-down menu contains various types of databases which are of primary, secondary or specialized type of databases. So I have discussed earlier all these database types in my previous video. If you want to see this video, check out the video link provided in the description. Now let's move ahead. Search for the nucleotide. Here you can see, click on this nucleotide and type the name of the gene of your choice. For this particular example, I have chosen the BRCA1, that is human breast cancer antigen gene, because it has been explored extensively for breast cancer research. So I am typing BRCA1 Homo sapiens and hit on the search button. In a few seconds, new window is opened and you will see that your search has rendered 4,496 results. For this particular example, I will choose the ref SCQ sequences, and specifically we will be studying on the NM_007294.4 gene. Hit this link and you will be moved forward to another page where you can see the human breast cancer antigen 1 DNA repaired associated mRNA sequence. Moving on to this page will provide you a plethora of information. Let's learn about these one by one. The first option here is the locus of the gene or location of the gene in your DNA sequence. So it has been given a specific numbers. This is basically alphanumeric input, and it is also called as your accession number or your version number. The version number specifically is different, that is 0.4, and there are various type of version numbers, that is 0.1, 0.2, 0.3, 0.4 and so on. The 0.1 refers to the very first version when the sequence were first deposited onto this database, and if it is revised time and again, then your sequence is provided the corresponding version number. So .4 here represents this has been the fourth instance of depositing this sequence. Moving ahead, you will see the number of base pairs in this gene, and of course it is an mRNA sequences and having a linear type of nucleotide sequence, and this word PRI states that this sequence is originated from primates, and here is the date when it was submitted to NCBI. And the second line, you are provided with the definition of this particular sequence. Moving downwards, you can see the organism or the source organism in which this gene sequence or the RNA sequences has been identified, and below you will find very redundant information like the author, the title of the publication, the journal in which the information about this RNA sequences has been published, the authors, titles, and so on. So, however, this information is not very important to us. Moving downwards, you will see your gene sequence in Genbank format. The Genbank format contains various rows in which each row there are 6 groups, and in each group there is total 10 sequences. So in the very first row you will see a total of 60 nucleotides or 60 base pairs. Likewise in the second row, which starts from the nucleotide number 61 and it ends at nucleotide number 120. For bioinformatic purpose, Genbank format is not useful because it contains gaps in between various nucleotides as well as numbers, which are a sort of limiting factor. However, for practical purposes, it is required to transfer or transform these sequences in a FASTA format. So for this, just click on the link send to option and select the gene features option, and herein you will see the option called as FASTA nucleotide, and just click on the link or the button called create file, and you will see that in a very few seconds your desired sequence is stored or downloaded in the download folders. You can just move to the download folder or the desired folder, and clicking on the link will provide you the FASTA sequences. So whenever you access the FASTA sequence, you will see it begins with a greater than symbol. This greater than symbol is highly important whenever you perform any bioinformatic applications. Below this first line, you are provided the gene sequence in a continuum format, and this particular format is of high utility in bioinformatic exercises. So that's how you explore the nucleotide database for identifying a gene and downloading the sequence of the gene of interest in a FASTA format. If you want me to create a tutorial on how to explore this particular gene or any other gene using BLAST, or how to align this gene with multiple other sequences, then please let me know in the comment box. If you like this video, then please don't forget to hit the like button and subscribe my channel, and also please don't forget to share this video with others. I'll see you in another video. Thank you so much for watching this video.